Good afternoon, everybody. My name is Stephanie, and I will be monitoring the event today. Just a couple notes. I will be sending out a Qualtrics evaluation towards the end of this talk, so please take the time to fill it out. We really value your opinions, and we'd love to hear different topics that you are interested in learning about in the future. Our next Leaders Initiative seminar will be Monday, February 24th, and Dr. Florence Johnson will be presenting the use of community support services and mental health among Black family caregivers. So completely different topic from what we're hearing today. So we're happy to have a wide variety of speakers come in. And with that, I will turn things over to Courtney for our introductions.
Great. Thanks so much, Stephanie. And thanks everyone so much for joining us today. So we're excited to have Dr. Arthur Zhou as our speaker for this month. Dr. Zhou is a research assistant professor in the Department of Computational Medicine and Bioinformatics at the University of Michigan. His primary research interest is the discovery and analysis of structural genomic variation within human populations and its impact on health and disease. So with that, I'll hand it over to Dr. Zhou.
Thank you, everyone. Thank you, Courtney and Stephanie. Let me share my slides real quick. And have a good. All right. Yeah, so thank you so much for having me here. And also, I know today is the Martin Luther King Day, so I really appreciate everyone come here for listening my talk. My name is Weichen Zhou. I'll just give a very brief introduction about myself. I am a research assistant professor right now in the Gil Oman Department of Computational Medicine and Bioinformatics. My primary research interest actually focus on the discovery and analysis of genomics and genetic variations in human population, as well as the impact on the population diversity and various genetic disorders. So basically, by combining my expertise in the bioinformatics pipeline with the data from wearable genomic technologies, we actually can explore the origins, distributions, and the mutation process of the genomic and genetic variants in the health and the disease samples. Today, I'm going to talk about one of the very interesting crypto genetic variant we called nuclear mitochondrial DNA insertion that we have some very recent interesting observations and in the human brain and other cell lines. Just a little before I introduce the project, probably I just a little bit step back for the background. Mitochondrial DNA. So as we all know, that mitochondria is the energy of the factory of the cells in human, and the dysfunction of the mitochondria actually will rise the oxidative stress, and it will link to it is thought to link to the aging process and the aging related diseases. Here we have in human genome, we have human cell, we have mitochondrial DNA that include a 16 base pair nucleotides and encode 13 proteins. And today's talk is mainly about the genetic variant, genetic element that we called NuMite, which is basically represents the nuclear mitochondrial DNA insertion. And what we see here, basically, as you can see, we can observe actually we will have a lot of the mitochondrial DNA in the mitochondria, and there by some chance we observed that phenomena that this empty DNA will transform into the nuclear, and inserted by accident or by any unknown mechanism inserted into the nuclear genome. And after we observed this, we cut this sort of small segments of empty DNA into the nuclear genome as a NuMet. And this incorporation of mitochondrial DNA into nuclear genome actually is a widespread phenomena in many species. And there are some studies have been reported that the NuMets are related to the cell function and also be observed in a lot of different cancer diseases. The reason we are interested is because like in the recent consortium we called Somatic Mosaicism network, actually we observed a lot of different somatic mosaicism genetic mutation or of the genetic variants that occurred in the human brain in different regions of brains. And also this sort of like somatic mosaicism would lead to the potentially lead to the development of brain and the neuron cells, as well as could potentially related to the neurodegenerative disease and for example, Alzheimer's disease or other psychiatric disease, neuropsychiatric diseases. There are many already in among these decades. Actually, there are many studies already reported the certain specific single nucleotide variation or copy number variations that happened in the specific regions of brain, for example, the hippocampus or frontal cortex, that could contribute to the progression of the some sporadic cases of Alzheimer's disease. And the question here, we were just wondering, like, are the NuMets a possible mitochondrial DNA, nuclear DNA signal access in the human aging and aging-related neurodegenerative disease? And especially we're focusing, since we focus on Alzheimer's disease, we would love to see whether they have any relationship. So our hypothesis here is that the basically the existence of somatic mutation could be correlated with the gene expression and regulation, while the cis and trans effect in the different brain regions or age groups or cognitive status. And the somatic mutation will accumulated with age in the brain or certain brain regions eventually would have the potential roles and the impact on the AD or AD-related pathogenesis. What we're looking at, the data set right now here is called the RoseMap cohort. It is archived right now in the NIAGAS database, and with all the very good harmonized data, clinical information matched to the to each samples. So here's the sample that in our project. Basically, we have more than 1,000 samples from the RoseMap data and from different individuals, and those are like the samples from the post-mortem brains and across different brain tissues, as well as some of the circulating immune cells. We also have collected the different information from for the samples, including the cognitive status. For example, like I show here, the different like the for example the non-cognitive impairment and mild and Alzheimer's disease status. and also the different age of the patient at death. With all of these samples, the Rose map actually have the whole genome sequencing for all of these 1,200 samples, and we are developing our pipeline to basically identify the somatic mitochondrial DNA or new mites in the nuclear genome. What do we do here? Basically, by obtaining the whole genome sequencing data, which is stories, and we have our own developed software named Dynamite from the previous study, and we extracted the signal from the of the mitochondrial numeric sequence or mitochondrial sequence in the nuclear genome, and then compare the ones that in the Rose map to the 1,000 genome project as a healthy control population, to do some cross-referencing analysis, and then finalize the tissue-specific new mites in each of the tissue from for the Rose map samples. This is basically a very brief idea of how we detect that from the whole genome sequencing data, just in case that some audience not familiar with the genomic content. So basically, we have the pair and the reads, and supposedly this is the reads that map to the reference genome, and if there is any mitochondrial sequence that in the sample will map to the mitochondrial DNA sequence. And when we see it, we will find some signal that one end map to the reference genome, one end map to the insertion or the mitochondrial DNA insertion. And by extracting those kind of those signals, abnormal signals from the sequencing data, we're able to resolve this mitochondrial DNA insertions in the nuclear genome. Okay. Okay, so here's some observations or results that we got. So first of all, as I mentioned, we have different brains, regions, and different tissues for the Rose map data. So the first question we ask, we check here is basically the whether there is an enrichment in the different regions of the brains for the somatic new mites. And actually, yes, we do observe a very high enrichment of the somatic new mites in the PFC samples. It's about like more than 5-fold compared to the cell membrane and like two other whole blood and the PBMC samples. And we further asked the question, does the new mite distribution correlated with age or cognitive status? Oh, sorry, my slides just got stopped. Okay. Here's the distribution here. So basically, the first this figure shows the genomic distribution, whether there would be the insertion bias in the preference in the genome. So we do observe that some of the tissue-specific new mites preferentially inserted into intro and intronic or intergenic regions, which makes sense because I bet most of the somatic insertion would be a mutation would be a benign or non-function. And we also look at the cognitive different groups for the tissues and the cognitive status as well as age groups. We do not see any stratification or difference between the different cognitive status or different age groups. Okay.
Can I ask a question?
Sure.
in an individual brain, can you determine whether there is a specific pneumite in, let's say, the dorsolateral prefrontal cortex, but not in the cerebellum? That is, can you do a region comparison within individuals, or is this a sort of a group assessment?
This is actually, yes. So basically, in our pipeline, we do cross-reference the pneumites in different regions of brain. So if there is an overlap between the pneumites, there's the same pneumites between the frontal cortex and the cerebellum, we would not consider as a tissue-specific pneumites. Right. So how about the other tissues? Sorry. What I have here is basically okay. So the next step, we want to check the age and death correlation with the different tissue-specific pneumites, and to see whether there will be some correlation, relationship here. As you can see the figure here, actually we observed a tissue DLPFC specific pneumites which are enriched, that as we did observed before, and negatively associated with the age and death in all the samples that we have. And we do not observe this significance in either the cell membrane or PCC or other tissue. And we further break this down, the tissue-specific or the DLPFC-specific pneumites into the different cognitive status. And we do observe the difference between the different groups of cognitive status. We observe the significant negative association in the non-cognitive impairness group, a very mild, almost like no significance in the mild cognitive status, but no significant correlation in the Alzheimer's disease group. And this is about, like, for the non-cognitive status, is about like 2 more pneumites accumulation per decades in those samples that with no cognitive impairments.
I'm sorry, it looks to me like it's not accumulation as a function of age, but the reverse. Am I right?
Yes.
That is more.
Yes. This is a negatively. So it's basically like if you have more pneumites in the non-cognitive groups, you will die early. Does that make sense?
Yeah. So I'm just so I understand. These are DLPFC specific. They're not found elsewhere. There are more of them in cognitively unimpaired individuals if you die younger.
Yes, exactly.
Okay.
Yes.
Okay.
Yeah. Yes. All right. We also look at the other, besides the DLPFC, we'll also look at the other tissue-specific pneumites and in the different cognitive groups. And for example, here, the cell barrier, we do not observe the very significant correlation for each of the group, although it's like under 0.05, but we do not consider it's a very good significance. And basically, this evidence has shown a connection between the Alzheimer's disease and aging and pneumite activity in post-mortem brain samples, but actually demanding us a in-depth investigation for while the range of data samples and extensive details. As you can see here, probably is the reason that we do not have enough samples and do not grasp this power to have the results. So more recently, we asked we tried to figure out, like, seeking for more samples, and also we try to implement the new technology for more accurately detect the somatic mitochondria. One of the technology that we are using actually is based on my previous work 2 years ago, that this is for the transposable element, but we do adopt this into the mitochondrial DNA detection. Basically, it's a new technology assay that we use the Cas9 guide RNA sequence that targets the specific mitochondrial DNA sequence. Well, here we show the MEI, which is the transposable element, but you can also replace this with the mitochondrial DNA sequence. And then with the Cas9 technology target on the mitochondrial DNA in the nuclear genome, we will put it out and cut it into bi-direction, and then sequence with ONT next Oxford nanopore long reads. With that, we can able to resolve not only the nuclear genome, nuclear mitochondrial DNA, but also we can determine where the nuclear mitochondrial DNA insert by using the flanking region information supported by the ONT reads. So we use this actually to do some pilot experiment. Here's two examples that I'm very exciting. It's just a very recent progress. Here, the first one is that we use this 10-Cas technology that targets the NUMA insertion, and we do show that here, as you can see, this is the NUMA insertion signal with the soft clip, colorful soft clip in the ONT reads from 10-Cas, and we can successfully capture the germline, at least the germline mitochondrial DNA insertion in the nuclear genome. And this figure here is not from the 10-Cas, but also very interesting to me is that basically we have developed a new technology, to sequence the single cell DNA with long read by using the Malbec DNA amplification. And in the cells, actually, individual cell DNA genome, we do observe a somatic NUMA insertion that are not observed in the book ONT genome sequencing, whole genome sequencing, as well as in the other reference 1 kg healthy population controls. And it is actually can be observed in 3 cells out of our 100 cell pilot experiment. So all of this experiment basically showing a very promising results that we are able to capture the somatic mitochondrial pneumats in the human brain, especially specifically here is the frontal cortex region, and we are planning to deploy this technology into the new samples after we do a better refinement of all of this the two technologies here. All right. So the other half of the story, I will probably just go very quick. Since we observed the PFC of specific mitochondrial insertions, nuclear mitochondrial insertions, are negatively associated with age at death in person, without cognitive impairments. Now we're looking at more potential correlation between the pneumatics and the aging process. And we have the collaborator in Columbia University we have here, basically they build up a longitudinal repeated measure WGS study, a whole genome sequencing study for the primary human fibroblast group under certain conditions with the medicine stimulation. And for that, So basically, this is the experiment design. We have 3 donors and fibroblast cell lines, and we basically culture them with from day 1 to day 200, and we have different treatment for these donor cell lines. Basically, with non-treatment or the dextro or oligo, which are the simulators like to mimic the oxidative stress in the environment, in the cell environment. We also have 3 patients that have the SIRF1 gene defect, which is the SIRF1 gene actually is one of the most frequent cause of the cytochrome oxidation and will lead to the mitochondrial disease in the human. With all the data points that we have, so basically we in each data points, and we extracted the cells and the nuclear genome DNA, and we do the whole genome sequencing. And we use the same pipeline to look at the somatic mitochondrial DNA that we used in the roadmap data cohort. And here's some results. So basically, we do observe a mitochondrial DNA accumulation with the days of culture in the cell line, meaning that basically the more age or more days they culture, the somatic mitochondrial DNA accumulated in the cell lines with the aging process. And the as a computational biology in the genomics, so we will ask a question whether those are just like some the same accumulation or the trend of genomic instability while the cell culturing, right? So we also compare this insertion accumulation trend with the other genomic instability factor, like the other genetic variants, for example, the deletion or duplication or inversions that in the same WGS data. And we do not find any of these genomic deletions or genetic variations that have a same accumulation trend or plot compared to the mitochondrial new mite insertion. And furthermore, we look at the different treatment of the mitochondria of these different samples, and to see whether the mitochondrial accumulation would have difference here in the different treatment or in the healthy donor versus the SIRF1 patient. And we do first of all, we do observe a graduate accumulation of 1 new mite per 9 days in the cell culture in the control donor healthy sample, no treatment. And we do the difference between the non-treatment versus the dext and the oligo, and as well as the SIRF1 patients. And if we compare those different insertion or accumulation rate, we do observe a increasing trend here. Basically, the SIRF1 would have very significant, SIRF1 patient cell line would have a very significant mitochondrial DNA new mite accumulation in the nuclear genome versus the non-treatment healthy donor. And it's about like more than 4-fold increasement here. So basically, our observation indicate that the mitochondrial or the new mite could be introduced by the homeless mitochondrial DNA outside of the nucleus, and the amount of this DNA mitochondrial DNA can be increased by stimulation from maybe the environment, cellular environment, for example, some stress or oxidative stress, or as in our project, the oxidative simulators, and or the genetic factors will also increase the chance of broken mitochondrial. Mitochondrial release segments of mitochondrial DNA into the cellular environment and can travel through the pores of the nucleus and integrate into a cell chromosome. That's our thoughts. And with that, higher mitochondrial DNA would have the more chance to integrate. But the mechanism of how the mitochondrial DNA into the chromosome is still unknown. So that could be one of the potential direction of our future project. All right, with all of that, I give some summary here. So in the roadmap cohort, we found the majority of the mitochondrial aggregated or enriched in the DLPFC regions compared to the other tissue, brain region or other tissues. And we further found that in the non-cognitive impairment samples, there are about 2 more new mites per decades of life lost than those have lived older ages. And the investigation of the new mites in a longitudinal repeated measured WGS study further revealed that the accumulation of a new mite of every 9 days of life independent independent of large-scale genomic instability. And we do observe that with the SURF1 mutation or the oxidative medicine, oxidative stress medicine that would potentially increase the mitochondrial DNA transferring from the mitochondria to the nuclear genome. And this is this work recently published in the biological PLOS biology, and actually it draws a very good attention by the public. We have a very good download in as the publication online, and it has many media just report this our work here. So with that, I would love to thank the collaborators in the Columbia and also in the DCMB, as well as in the Rush University. And then I also would love to thank the MADC mentorship team and all the MADC leadership. Thank you so much. I would take any questions. Thank you.
Arthur, this is Hank. Thank you so much. It's really exciting work, and congratulations on the popularity of your paper on new mitogenesis. You know, the most striking finding you have is the inverse relationship between new mites and cognitively unimpaired individuals in age. But, you know, a lot of interest in your article is going to have to do with aging and dementia and the idea that there's a mitochondrial cascade that might actually drive some aspects of dementia. What can you say about, and the dorsolateral prefrontal cortex is very important in Alzheimer's and in FTD. Do you have a sense of the relevant importance of mutations in nuclear mitochondrial genes and its relationship to dementia itself?
Actually, that's a very good question, because we are thinking this for a long time as we are working on this project, and especially with the results that I just showed. Because we know that actually the for example the other genetic variant, for example the transposable element and some deletions and single nucleotide mutations that have been reported in the publications or observed in the DFPFC regions of the human brain. And the mitochondrial DNA or the pneumite, we will say it's more like a indicator or the results of the whole chaos status of the cell, either in the in the healthy control or healthy population, or in the dementia patient. I would say, like, as you know, like as I showed that with the different oxidative stress stimulate, and also the genetic defect cell line observation. Actually, we think that the mitochondrial insertion, somatic mitochondrial insertion in nucleus would be the indicator in the generally in the samples. But speaking of the dementia patient, I would say we do observe some of the slightly significant correlation, just correlation between the somatic pneumatics into the mild cognitive status samples in the DFPFC, and also the Alzheimer's disease samples in the cerebellum. I would say the reason why this weak correlation could be, like, we need I would say, like, first of all, could be the detection method. Basically, we're using the short-read sequencing technology for that, and the read depth is not very high, and it would limit our detection of the somatic pneumatics within these samples that we don't know. And also, like, the sample size is not that big. So I would say that's why I introduced some new technologies, and, like, so basically the motivation that we really want to dig into, like, whether those somatic pneumatics are really exist in the different brains of regions of the dementia samples or Alzheimer's disease samples. Yeah, right now I really don't know. For, like, just to summarize what I said here, I basically think that the pneumatic is an indicator, not a really would not cause cause the disease, or, like, yeah.
I have other questions, but if other people want to ask a question, I will pause. All right, I'll ask another question quickly. Most of your mutations are in the introns. There's a lot of interest right now in intronic short tandem repeats that expand in aging and in disease states, and that they may be contributing to Alzheimer's or other dementias. And some of those short tandem repeat expansions could actually lead to RAND translation, the production of these weird products. And there's a paper in press in PNAS showing the accumulation of RAND translation products derived from short tandem repeat expansions that are CG rich. That's a long introduction to say, your mutations, are they mostly missense changes? Are they changes in the length of STRs? What are they?
That's a very good question. Actually, we did not look at the genomic insertion content of the somatic pneumatics. I would definitely look at this now. That's a very good question. And also, like, since we have checked the accumulation of in the cell, this is not in the brain, this is just the agent cell line. So we check the different genetic variants, like the duplication or the insertions of the cell line, and we do not see accumulation trend here. But on the other hand, for the short-read sequencing data, we have the limited power to detect the STRs. That's the point. So that's why like really like we want to use the long read sequencing to try to figure out whether there would be some short read tendon repeats expansion or contraction within the samples. Yeah.
Sounds great. And also relevant to your transposable element work for sure.
Yes, yes. And also just one more comments here. Very interesting. There is a subfamily of transposable element which have a lot of GC content region as small tendon repeats within the transposable element, and we do want to look at this whether it will contribute.
I would love for you to look at that. I would love for you to look at some of our own samples, so we can talk more.
Yeah, thank you so much.
Any other questions for Arthur today?
Thank you, Arthur. Really beautiful work. Congratulations.
Thank you so much. Thank you all.
All right, well, thank you all for coming out today. We'll end things a little bit early. Be sure to fill out the event evaluation, and we'll see you on February 24th for our next Leaders Initiative talk. Thank you so much.
Thank you. Thank you, Stephanie.